Hi everyone, I'm Maxine. I'm a master's student in the John Parkinson Lab at the Hospital for Sick Children in Toronto. Today I'll be talking about our study on the performance of microbial-based solutions on a Campylobacter infection challenge in poultry. Antibiotic growth promoters, or AGPs, are commonly used in the poultry industry at subtherapeutic doses to improve feed conversion, bird growth, and mitigate infection. The exact mechanism by which AGPs promote growth is unknown, but it's been proposed that they operate through modulating the gut microbiome. One hypothesis is that AGPs exert this overall antibacterial effect, which may protect nutrients from bacterial destruction, decrease production of toxins by intestinal bacteria, and most importantly, it reduces the incidence of intestinal infections. And AGPs have shown to be effective in mitigating against Clostridium perfringens, and it may protect against Campylobacter and Salmonella as well. There's been a rise in pathogens developing antimicrobial resistance mechanisms, and it's been linked to the use of AGPs. And since these mechanisms are such a threat to public health, it's expected that there will be a global ban soon. But without the ban and without alternatives, enteric infections are expected to increase, and this would hurt sustainability in the industry. So there's this interest in using microbial-based solutions instead, for example, single-strain probiotics and microbial consortia. Single-strain probiotics have shown promising results as a growth promoter. They have a transient effect, and they need to be constantly administered. Whereas microbial consortia is defined as a small collection of bacterial strains that represent an adult chicken's microbiome. And it's more stable than single-strain probiotics since it's able to engraft more readily in the microbiome due to the mutualistic interactions between the bacterial members. For example, single-strain Saccharomyces cerevisiae boulardii has been shown to reduce Campylobacter and Salmonella, and Avogard has been successful in mitigating against Clostridium perfringens and Salmonella typhimurium. For a probiotic product to replace an AGP as a growth promoter, it's key for it to be able to mitigate against infections. And a lot of the interest has been focused on the ability of these microbial-based solutions to protect against Clostridium perfringens, which causes necrotic enteritis, which can kill whole flocks. But there is a gap in knowledge on Campylobacter jejuni, which is a gram-negative bacterium considered to possess a near commensal relationship in chickens, but when exposed to humans, it can make us very sick. It is a leading source of foodborne illness in industrialized countries. With our collaborator, Dr. Cheyenne Cherry from the University of Guelph, we looked at 4 different industry probiotics products to bacitracin, which is an AGP that's commonly used, and we compared their efficacy against a Campylobacter challenge. And we used 2 single-strain probiotic Saccharomyces cerevisiae boulardii and Pediococcus acinetobacter. And we also used 2 microbial consortia, Avogard and MCH1. MCH1 is still in development. The animal trials were done by Khaled Abdelaziz from the Sharif Lab. And Khaled took newly hatched chickens on day 1 and separated them to 8 groups. Avogard and MCH1 were given as 1 shot for the first 24 hours. Whereas all the other treatments were given throughout the experimental period. And untreated chickens were fed a normal diet and were not given any treatment. On day 14, all the groups, apart from the 2 control groups, were challenged with Campylobacter. On day 30 and day 39, chickens were sacrificed and samples were taken from the ceca, which is a pouch that starts the large intestine. So at the end of these trials, we had around 10 chickens per treatment per time point, a total of 158 chicken cecal content samples. These samples were sent for 16S rRNA sequencing and also plated onto Campylobacter selective media and incubated to quantify Campylobacter count to colony forming units per gram of cecal content. Here, Khaled compared the mean of Campylobacter counts on a log of 10 scale on many different treatments, and we found that none of the treatments were able to reduce count at day 30. But at day 39, we found that treatment with the Pediococcus and bacitracin failed to reduce Campylobacter count. And the non-significant increase of Campylobacter count by bacitracin may be because bacitracin targets gram-positive bacteria, which may have opened up a niche for the gram-negative Campylobacter to further colonize. And we found that treatment with Avogard and MCH1 and the Saccharomyces were able to significantly decrease Campylobacter relative to the bacitracin samples. It's also been found that Avogard and Saccharomyces have been shown to decrease Clostridium perfringens colonization, so perhaps replacing bacitracin with either of the two microbial consortia or probiotic yeast may be more successful in reducing both Clostridium perfringens and help mitigate against Campylobacter. As I mentioned, chicken sequel samples were submitted for 16S ribosomal RNA gene sequencing, and then I processed these reads and clustered the sequences into OTUs, or operational taxonomic units, which is basically a term used as sort of proxy for a microbial species where the taxonomic assignment is still unknown. Basically, you align or map these OTUs to a database of known RNA sequences, and basically you're able to reconstruct the microbial community at the end of this process. So these steps yielded a total of around 2 million reads, which were clustered into 1,305 OTUs. Then I use a combination of R programs for the following downstream analysis. And in doing so, I wanted to see the effects of each probiotic or microbial-based treatment on the microbial composition and compared to the samples that were either untreated or treated with the AGP. Next, I wanted to see how different treatments affected alpha diversity in the microbiome. Alpha diversity is a term that refers to the diversity within the microbiome. High diversity in the microbiome usually means it's a healthy one. There are two things that contribute to alpha diversity, which is species richness and species evenness. Species richness refers to the number of bacterial members or OTUs observed. Species evenness refers to how evenly distributed the bacterial members are in the microbiome. So you need to take both of these into account when interpreting the overall alpha diversity in the samples. Just an example, both of these microbiomes had the same 4 unique members, but microbiome 1 is more evenly distributed than microbiome 2. So microbiome 2 would have a lower species evenness. Here, the number of bacterial members found in each treatment groups are compared in this graph. The black points belong to 30-day samples, while the gray points belong to 39-day samples. And we noted that treatment with the single-strain Pediococcus had the highest number of bacterial members. And this graph compares how evenly distributed the bacterial members are in the samples between the different treatment groups. A higher score means the microbiome is more evenly distributed. And we know that treatment with the single-strain Pediococcus resulted in a microbiome that was the most evenly distributed compared to all the other groups. Compared to other treatment groups under challenge, samples treated with Avogard had the lowest number of bacterial members. And we also found that these samples had the least evenly distributed microbiome relative to all the other treatment groups. And this overall lower alpha diversity may be because the small collection of bacterial members that make up Avogard can quickly form a community that prevents other bacteria from growing. Assigning the 1,305 OTUs revealed unique microbial signatures associated with each treatment and time point. The taxa and the legend is listed in order of appearance. And we found that there was a higher relative abundance of Bacteroideaceae family within the challenge samples treated with Avogard. And at the genus level, these reads were assigned to bacteroids. And we also found a high relative abundance of Rickenellacea and MCH1 samples. And at the genus level, these reads were assigned to Allistipes. And both Bacteroideaceae and Rickenellacea belong to the Bacteroides phylum, which it's known to contain bacteria that are major producers of short-chain fatty acids like acetate, propionate, and succinate. And this phylum is also considered to be a stable part of the chicken gut microbiome. And we found that the Pediococcus treatment had a transient effect. No reads were actually assigned to Pediococcus. in samples treated with Staphylococcus, but we do see this abundance of bacteroids and Bacteroideaceae and Organelles in the 39-day samples. And as our sequencing only targeted 16S rRNA, we weren't able to observe the ability of Saccharomyces to engraft within the microbiome. To summarize, for a probiotic to replace an AGP, we need to be able to mitigate against infection, which we saw Avogard, MCH1, and Saccharomyces do. And then I wanted to see how the different treatments would affect the microbiome composition. And we saw that single strain Staphylococcus was unable to engraft into the microbiome, but we also saw that it actually promoted diversity. And we wanted to see if any of the probiotics promoted any beneficial bacteria. And Avogard and MCH1 was able to do this consistently across time points. Overall, taking into account all these factors, no one probiotic had better results than the others. So depending upon the farm and the flock, they can choose any of these probiotics to better suit their needs. Thank you for listening.
Thanks for your presentation, Maxine. We've already got a couple questions here, so I'll wait for your video to come live. Hey, there you are. I hope you can hear me all right. It sounds like my Wi-Fi is
I can hear you. Yeah.
Great. Okay, so let's go with the first question from Sasha. It says, in the first part of your study, in the efficacy of the different products, the variation in the CFUs seemed very large. Do you know why, in general, the CFU of Campylobacter between individual chickens differs so much?
Individual chickens.
Do you know why, in general, the CFU count of Campylobacter between individual chickens differs so much?
Oh, well, it can be a number of factors. These chickens aren't all, these chickens aren't purebred. They're not all the same chickens. They have individual, and we also chose a small sample size, around 10 chickens per group. So with a small sample size, you do have the chance of capturing those outliers.
Good. And there's another question. It's just a comment that says Campylobacter jejuni does not cause any symptoms in chickens. So how can you tell that your challenge was successful?
So we plated these samples on Campylobacter selective supplement, and we also looked at the sequence counts for Campylobacter. And in the untreated groups that were not challenged, we didn't see any sequence counts assigned to Campylobacter, and we also had a negative result for the colony forming units.
Okay, so our next question is, did you see differences in the Campylobacter family with the 16S data analysis?
Oh no, we only picked up on one strain of Campylobacter.
Okay, and then I'm just going to go down to here. Were the chickens housed in cages or on the floor? And do you think that this would make a difference in your study?
They were separated into different rooms, so it would prevent chickens from eating other chickens' feces. So that would make sure that there's no sort of sharing with the microbes that are from the environment.
I think the question was kind of asking whether or not they were in cages or on the floor, like whether or not they had access to feces or not.
Oh, they did have access to feces, but these chickens in different treatments were in separate rooms.
Gotcha, good. Okay, and then your last question is, what is the challenge dose of Campy to establish the model? It seems that the CFUs per gram and fecal contents varied largely.
It was 10 to the 7 colony forming units that were introduced.
Good. Well, I think you answered all your questions really well, and you got a lot of them, so that's exciting. So thank you very much for your presentation, Maxine.